Hi, 11s and 12s. We're moving into the second week of looking at cells now. We're going to start by looking at a little bit more at the prokaryotic and eukaryotic cells in detail. We've looked at them very briefly last week, but this week we're looking in a lot more detail. We're going to start though by talking about microscopes, because when we're talking about cells, everything that we've learnt about cells, etc., has all started with the invention of the microscope. Without the microscope, we couldn't see anything like this, which is where we're starting here. So, look, a majority of progress in biology in general, because biology is the focus of life, and most of that is in the cellular level. So a majority of the progress in biology over the last 150 years has been restricted by the improvements that the microscope sees. So looking at the second half of the 19th century, inventing the light microscope helped us to discover bacteria and unicellular organisms that before we had no idea existed. Very soon after that, we've discovered that we had chromosomes, mitosis, meiosis, all of these being observed for the first time. The whole idea of cell division completely eluded people until we had the light microscope to be able to see these objects and actually visualize what was happening. The problem with light microscopes, of course, is they cannot produce a clear image of objects that are any smaller than 0.2 micrometers. Remembering that a micrometer is 1,000th of a millimeter, and a nanometer is 1,000th of a micrometer. So light microscopes, anything smaller than 0.2 micrometers, really hard to see. So in the 1930s, light microscopes were developed in Germany, and they were used in the 40s and 50s. These helped a lot, obviously improving the clarity, letting us see more detail. But then when we got the electron microscope, that's helped really reveal the structure of cells, because the electron microscope is so much more refined, allows us to see at a much more clearer level all of those small objects that we just can't see with a light microscope and with the human eye. So, talking about the resolution of electron microscopes, I suppose first we start with some definitions. So, when you're seeing something and you're making something visible and distinguishable by the eye, we call it resolution. So the maximum resolution, the amount that you can see through the eye with the use of a light microscope, is 0.2 micrometers. It doesn't matter how powerful the lens is. The highest resolution is going to be limited by the wavelength of light, because everything we're seeing is based on the wavelength of light. That means that the maximum lens of a light microscope is usually 400 times magnification, because we're not going to get much more clarity or more resolution going beyond that. Whereas we look at electron microscopes, the resolution of the current electron microscopes are 0.001 of a micrometer, or basically, we'll talk about nanometers in a second. So 0.001 of a micrometer, and that's because we're not using light to view objects here. So what's happening with an electron microscope, as you can probably guess by the name, it's a beam of electrons being fired at an object, and it bounces back off the object, back to a sensor, and it determines the surface of something, and we can see a lot more detail because of that. Now, because obviously we're using electrons and we're not using light, that means the electron microscope images are generally going to be in black and white and not colour. The ones that you do see in colour have generally been edited post-image to add the colour to it. And I'm just finishing off with this video, looking at a little bit of what can I see. This is from a website called BioNinja that kind of summarises what we can see with different microscopes in our eye. So you can see with the human eye, we can't really see anything smaller than 100 micrometers. When we go with a light microscope, you're really not seeing anything larger than 100 nanometers. With an electron microscope, you're not seeing anything smaller than 0.1 nanometers or 0.001 micrometers. Got the idea here. With the eye, we can see, for example, hair, but we're not going to be able to see blood. With a light microscope, we can see hair, blood, and bacteria, but when we get to things like viruses, DNA, glucose, and atoms, that's just not happening with a light microscope. You need an electron microscope to see those things. And there's even some bacteria that are so small that you won't see with a light microscope. We've got quite a few pathogens that we can look at under the microscopes when you come back to school, and you won't be able to see much. You can see some detail, but it's really hard to work out the individual cells, etc. Look, that's it for microscopes. Next video we'll move on to prokaryotic cells.